So, anyways, the bottom line is what we're going to do is I'm going to start by reviewing photosynthesis and respiration. Make sure you get the key points that you're going to need to do well on any question you might get on the OST or in on your final. The other thing is from there, I would like to review anything that you want. If you don't have anything to review, I'll just start from the beginning. Okay? All right, so we have two processes that we're talking about here, and I'm going to try to put all the ideas together. And so you have photosynthesis, and we're going to actually talk about three processes. Chemosynthesis, and you're going to talk about respiration, cellular respiration. And here we're talking about anabolic metabolism. Here we're talking about catabolic metabolism. This one, consumers use. Consumers use only respiration, where producers use both. All right, so producers use both respiration and one of these, while consumers only use cellular respiration. Now, because respiration is a little closer to because you just did it yesterday, you just finished it yesterday, let's talk about respiration first. Respiration can be broken down into two types. So you have aerobic. So you get aerobic or anaerobic. Aerobic has to do with air, and anaerobic, no air. And it's not actually air, obviously, at least I hope that's obvious. It's, I should say, let me get rid of air. I just like to use air because aerobic, anaerobic, but I'm going to put air / oxygen gas, right? So it uses oxygen gas, where anaerobic is no air, no oxygen gas. Okay? So aerobic respiration has to do is what happens when you use oxygen. Anaerobic air respiration is when there's no oxygen being used. We do both. Metabolism. In respiration in eukaryotes, this cellular respiration happens in both eukaryotes and prokaryotes. So in eukaryotes, it happens in the mitochondria. But because, and I keep saying this, oh my God, I hope it sinks into your brain, because prokaryotes don't have any membrane-bound organelles, they can't do this in a mitochondria. They don't have mitochondria. They have no membrane-bound organelles. So they only have, so eukaryotes, they do it in the mitochondria that looks, something like that. So in prokaryotes, they do it just in their cytoplasm. All right. They do, and when I say mitochondria, we're talking about aerobic respiration. The mitochondria does aerobic respiration. The mitochondria does aerobic respiration, okay? In the cytoplasm, prokaryotes can do either one, aerobic or anaerobic. So now let's break that down. Let's take a look at this respiration in a eukaryote. We're going to look at a eukaryote, and we're going to look at a mitochondria. All right, so in a eukaryote, obviously you have a cell. Both eukaryotes and prokaryotes are cells. You have a nucleus, you have a nucleolus, you have endoplasmic reticulum. I'm reviewing as I'm talking. You have ribosomes that are on the endoplasmic reticulum that would be part of the endopathic, or the pathway for proteins that are leaving the cell would be here. You have vesicles. You have a Golgi body. You have a Golgi body that has vesicles on it. They're fusing, and where the endoplasmic reticulum vesicles leave and move in that direction, then they leave the Golgi body and they travel to the cell membrane, where they fuse with the cell membrane and release their particles to the cytoplasm and to the outside of the cell. So these proteins indicated here in yellow, it could be in a plant, it might be a protein like capsaicin. But whatever it is, it's going to be produced in the rough endoplasmic reticulum, travels to the Golgi body, then travels out and comes out of the cell. Then there's ribosomes inside the cell, in the cytoplasm, that make proteins that are necessary for the cell. Some of these vesicles with proteins actually go and fuse with other organelles, like maybe maybe what's in here is hydrolytic enzymes, and they will fuse. This thing is then a lysosome, and they come from the Golgi body too, and originated from anything that's going in a vesicle, anything that's going inside a membrane, any protein that's going inside a membrane is going to be going through this endoplasmic reticulum. Any protein that's just made in the cytoplasm is just going to be used in the cytoplasm. So lysosome could be connected to a food vacuole. So here you could have food. It's a vacuole, it's a membrane. That came through something called endocytosis. So that's when your membrane's coming in. Instead of stuff going out, stuff has to come in. Food is going in, and has to be further broken down in these food vacuoles. This membrane used to be part of the cell membrane and just pinched off. Now you know this because you've blown bubbles before, correct?
Yeah.
You know that bubbles, what are bubbles? You've made bubbles, you blew them in the air, and they fuse together, don't they?
Yeah.
One bubble can touch another bubble and they become one. And sometimes they pop, and sometimes they split off into multiple bubbles, right?
Or you can blow, like, you ever seen a bubble with like 2 and 1?
Yeah, you can blow 2 in 1, or you can blow a bunch of them, right? So that's what's happening here, except it's not air blowing, they're forming because of chemical and hydrophilic and hydrophobic interactions. What's happening is that the membrane's folding in, and you get a bubble a bubble is a membrane. It's a single membrane. This is a double membrane, double lipid membrane that holds the food that's going in. This is again called, remember that word endo, because it's going in, cytosis.
Endo means enter.
Endo means enter, you can think of it that way. Endocytosis, so entering the cell. Cyto means cell. All right, so the process of going into the cell. This is called exocytosis, is exiting the cell, is correct. So for exocytosis, the pathway is going to be through those proteins and into a membrane. All right, so that's I think that's a decent review of that. But then we have this is really too big, but I'm going to go ahead and draw a little bigger than it should. Some of this is not to scale. And then you have like a membrane within a membrane. All right. So that's really weird, because in all the other organelles that are in the cell, none of them have two membranes.
What was that thing called? What, exocytosis?
Exocytosis and endocytosis. I'm recording it so you can catch up, but because we only have 2 days, I'm not gonna, unless you have a specific question when I'm talking about it, I'm not gonna go back. Is that okay? All right, so we have mitochondria. Now, it's interesting, mitochondria have this double membrane system. Let's look at mitochondria. Have two membranes. Mitochondria have their own DNA, which is really weird. Like, why are there two sets of DNA? Why do you have the DNA that codes for everything in your body except mitochondria? And then that mitochondria has DNA that's very much like prokaryotes. What do I mean by that? I mean, they're round. They have all the indicators of a prokaryote, of a bacteria. That's interesting. Isn't that interesting? Isn't that weird?
Yeah.
I mean, it's really strange. And then another interesting point is that they have ribosomes that are more like prokaryotic ribosomes. It's just really strange stuff. This mitochondria for you. So mitochondria, let me quickly go over it one more time. Two membranes, their own DNA. Remember, we get our DNA from our parents. So they have their own DNA. In fact, we can follow, this DNA only follows the mom. So we have followed what's called mitochondrial Eve. We found pretty much the first human. We went back all the way back into East Africa, because we can follow the DNA evidence all the way back. So it's interesting. This only follows mothers. You'll see why probably in January. Ribosomes that are like prokaryotic ribosomes. What's interesting also, though, is that there's a similar organelle that we've talked about that also has two membranes. Also, again, very rare for an organelle to have two membranes. Their membranes are organized differently, but still they have two membranes. They have two spaces in two compartments. Remember, that's one of the characteristics of life, that you compartmentalization is correct, and organization, right? So you have this chloroplast.
Is that a grana?
You know what's really, these are the grana, these are thylakoid membranes, they have the stroma, they have the thylakoid membrane. Mitochondria have similar aspects as well. But isn't it interesting that these characteristics that you find in mitochondria, they are also found in chloroplasts. Two membranes, their own DNA, different from mitochondria, but still like prokaryotes. Ribosomes that are like prokaryotes. Like, it's really weird. So scientists tried to figure out, well, why is it that these two really important organelles, right? Like, arguably, we could not exist without chloroplasts because we don't have them, but we need them to capture sugar, make sugar for us. Without them, we die. As most of you are sitting there falling asleep because you don't have enough sugar or you're eating sugar. Either way, like, you need this. This is an essential part of being alive. Being human is sugar, and it comes from these things we call plants. And yet, we were wondering, this was probably 10, 15, 20 years ago, 20 years, maybe, no, my God, I'm too old. More like 40, 50 years ago, people are like thinking, why? How is it possible that in every cell of our body, we have something that has different DNA from us? When we get something with different DNA in us, we call it a what? What do you call it when you get something in you that has different DNA? Not you, right? It's an infection.
Oh, yeah.
What your body? If I transplanted my kidney into her body, what would probably happen? Rejection. Your body, like, we were like, wow, this is really strange. Like, how does this? What is going on? So this is the theory. This is how we explain it. We have to explain it because that's what we do in science. We have to come up with a theory, and it's called endosymbiosis. I love the name. Endosymbiosis. Can I get my props, please? Can I get my props? I spelled it. I spelled it correctly.
Great job.
I will bow later, but let's move on.
First time and forever.
Yes. Endo means in. Symbio means to live closely. So symbiotic means to live closely with. There's several forms of the same word, right? Symbiotic, symbiosis, right? The same word, just different usage, different connotations, different aspects of the same idea. Living closely, right, to each other. Endo means in.
Enter, yeah.
In or enter. Right? In or enter. Are we good? So this theory explains chloroplasts and photosynthesis. Also, there's one other thing you have to think about. Which is also very interesting. This theory also explains that. Plants have mitochondria, but animals don't have chloroplasts. So we can look at the mitochondria of plants and go all the way back and find where the mitochondria, what beast the DNA is very similar to when we compare DNA. For all plants, we can follow it all the way back. Everything with a chloroplast. We can follow it all the way, even all the way back to algae, which are protists, single-celled photosynthetic organisms that have chloroplasts. We can follow the DNA all the way back in a chloroplast. And then the mitochondria in the plants, we can follow all the way back to a common ancestor with animals. Right? Well, we have. But let me show you what the theory is so you understand. How do we explain all this? Well, we explain is that we're thinking that there was a creature, single-celled creature. When I say creature, I don't mean monster. Right? It's a living thing. It's a living thing. So there's a thing, it was likely some kind of thing that had some membrane-bound organelles. It had DNA more likely than not, and it had some kind of, we don't know what kind of organelles it had, but it had some kind of organelles. What it didn't have was a mitochondria. And what it would do is when this thing found, food, it ate food, and it's sitting there, and here's this little,
How does it make energy? If that
it eats. So it goes through and eats, right? It uses anaerobic respiration, because anaerobic respiration does not require
ATP?
it doesn't require oxygen, and also doesn't require mitochondria. So everything here moves slowly. Without a mitochondria, you only have a little ATP, so everything's moving slowly, right? So, or maybe it does. Anyways, let's move on. So it's not very efficient. It's not very good at all at doing its job of living, right? It can't make ATP very well at all, but it does what it can. So it comes along and finds this little beastie it's going to eat. So the theory is that it gets, like Jaws, it gets closer to the beastie, the food. Just like Jaws. You know Jaws?
Yes.
Okay. I don't know. That was my generation. So I don't know. The shark, yeah. It gets closer, and as it gets closer, I'm sure the little guy was sweating it. It's going to do endocytosis, but it's going to do a specific type of endocytosis called phagocytosis. Oh my God, it's like dealing with little children. Phago means bringing large things in, right? So eating large amounts. So phagocytosis is bringing into the cell large things. So the cell is going to bring it in. It has to start to cave its membrane in, right? In order to be able to get it to surround. It's surrounding it. So it's going to surround it. And then finally, in one of its last steps,
It's gonna bend and absorb it, right?
it's going to have it. It's going to have it inside itself. Oops, why is that so?
It's like Pac-Man.
It does. So eventually what will happen is that the food will be inside,
It's floating in there.
and then, of course, as we know, we can have little lysosomes, right? Or something like it. Break down that food, right? And make ATP. Not very well, not a lot, but make ATP. That's called phagocytosis. That's how we absorb big things. So when you eat, you do phagocytosis in your intestines, your liver, and all that all the time, because when your white blood cells attack bacteria and you get acne, and you put all those stars all over your face, that acne bacterial infection is your white blood cells surrounding the bacteria and digesting the bacteria, killing the bacteria, because the bacteria is invading you. This is your army defending yourself against the bacteria. So that is how it normally works. But we think, when I say we scientists, that at one point in the history of life, something went wrong or went right. I don't know, it depends on how you look at it. Went wrong or right. It got swallowed. The bacteria got swallowed. The bacteria was food, but it didn't get digested. That's right. It didn't. This little guy got swallowed. Now it had its own membrane, right? And it was really good at respiration. It could make a lot more ATP. It was really good at respiration. All right. But it didn't get eaten. So it just stayed there. Now, it's still alive. It's in there. It's still alive. It has its own DNA. It's a bacteria, so it has all the characteristics of a prokaryote. It can do photosynthesis really well. The thing it couldn't do very well is move around. It couldn't eat big things very well. But the big cell can do it. So this thing is inside and lives closely with the bigger thing. What do we call that? Endosymbiosis. So they're living together. The big guy brings in the food. The little guy makes ATP. Fast forward a few million years, and now the little beastie is doing really well. The beastie's doing really well. Probably it evolved all kinds of different organelles. And the really cool thing is it has this is no longer a creature stuck in another creature. This is now a colony of little dedicated organelles. They can't live without the big cell. The big cell can't live without the big cell. They've come that close to each other. They can't live without each other. So now this little guy is making ATP, a lot of it, and and the big guy is getting sugar and pyruvate, and changes it to pyruvate for the mitochondria, and it's taking that pyruvate and putting it in all the mitochondria, and they're making all kinds of ATP for the cell. I don't want to make you think that the ATP is leaving the cell. It's all staying in the cell. So the ATP is just being made like crazy inside the cell by these mitochondria. ATP? Pyruvate? It's in your packet from yesterday, but it's a pyruvate.
Oh, yeah.
Yeah, it's pyruvic acid, it's pyruvate, yeah. So there we have it. So that's probably the first iteration, the first example of an animal ancestor. So this is where this guy will eventually, through many, many billion years, form the first animal.
What does it look like?
We have fossil impressions of these guys, but no, not these guys. The next one that I'm going to show you, they're called cyanobacteria. They would be the things that are inside. But let's move on. So one of these animals, though, one of these little guys did something even crazier. It happened again. So this guy had a mitochondria. It was doing well, making ATP. They were eating all these little beasties that were capturing light, making sugar. But they went ahead and did phagocytosis. And this time, though, they absorbed a little bacteria, a little prokaryote that could do photosynthesis really well. And the whole process went over again millions of years later. You have a beastie that has a mitochondria, all right, and that has these things that we call chloroplasts. All right, so we have chloroplasts, we have mitochondria. That over billions of years turns out to be what? The first ancestor of plants and algae, right?
Why didn't I go to animals?
Why did what?
Why didn't I go to animals?
Well, animals are the ancestor of animals did not have chloroplasts.
Why didn't I absorb it?
Say what?
I absorbed the food and stuff and made mitochondria.
It just didn't. It's about random. The theory suggests it's random luck. It has to be the right chemical combinations, just the right conditions for it to happen. If we're a village and we're out, some of us go hunting, and he doesn't find any food, and he and his family die of starvation, but I find food and my family and I survive. It's random luck. to some extent is also natural selection. So there's one, I was better fit to survive and he was less fit to survive. So him and all his genes die and all my genes survive. It's best fit to survive. It's just an example. We could have reversed it easily enough. All right. And so it all depends on the conditions. You might say, well, he's better fit, he's handsomer. That's not about what your idea of who's in better shape or who's handsomer is irrelevant. Best fit to survive just suggests who is better under these circumstances. In the days of kings and queens in England, it was the sneakiest, cheatinest scoundrel available was the king and the princes and the dukes and duchesses. They were the people that can maneuver, right? That can trick people out of their money. Like when you go to Wall Street, what's a good businessman? Someone who can collect money in a way. I'm only saying this to make a point, all right? This is not really true. But in a way, being a good businessman is being a good thief, right? It's a way of saying, legally saying, here, I'll give you a good example. Insulin is necessary for life. It costs the pharmaceutical company, I'm gonna make up a number, $1 to make. But the pharmaceutical company knows you need this to live. You're gonna pay me whatever it takes. So you know what they did? They did this. The guy got caught up in the government regulations, and I think he lost his job eventually. But they actually did this. They said, we're going to sell it to you for $1,000.
Why?
Knowing that a lot of people would die, but also knowing that a lot of people would pay that $1,000 and they would make a lot more money. So a lot of people would consider that immoral, not illegal, by the way, but immoral.
They're basically killing people off.
Yeah, it doesn't matter to them, right? They just want to make money. So my point is that they were best fit to survive. They survived. They're living in the best house. They can have 1 million babies, right? Where the people that are good, maybe they're honest, they're working hard, 2 jobs, they go to church, they do whatever, they're dead.
Oh.
They don't have babies. Sorry. They don't get insulin, right? Who's best fit to survive? It doesn't mean that you're handsome. It doesn't mean you're strong. It doesn't mean you're good. It just means you're Genghis Khan. Have you heard of Genghis Khan?
Heard, yeah, it's awful.
No.
Genghis Khan has his DNA in almost all of us.
Okay.
His DNA, we can trace Genghis Khan's DNA in almost, I think, something like some ungodly percentage of the human race.
Who is he?
Khans were great warlords of the plains in China. Genghis Khan conquered most of the known world. He conquered China.
Dang.
Do you know how he got his DNA into most of us?
He had babies everywhere?
Everywhere he went, he had babies with the women. He raped them. He was war. He found them, took them for their wives as slaves, raped them, had babies. Everywhere he went.
It was a white man?
No. Genghis Khan is Asian. Russian, Asian, like the Khans were in between in the Ural plains. So anyways, the point is not race or anything like that. It's that the man, you could not call him good, right? He was not moral, right? I mean, you can't call someone like that moral. But he was known as the Great Khan. Great does not mean good. Great just means powerful. And he was the best fit to survive. He had the most number of babies. Period. I have an ancestor who is an Irish king. But that's okay. There's 1 million of us out here. The reason there's 1 million of these people, and some of you might be, if your people are here for more than 6 generations, some of you might be here, might have his genes in them, because that Irish king had many babies, and their genes spread throughout the world.
Do you?
So many of us in the room. You might say, I'm not Irish, I'm not European. Some of your ancestors were slaves, and some of those slave owners were Irish, right? So, oops. Here's the thing. History is not about morality, right? There's a lot of immoral, wrong things that happen, and evolution is not about morality. Nature's not about morality. The hawk doesn't care that the rabbit's cute. It's going to swoop down and eat. It's going to tear its flesh apart and take its whatever flesh it can to its babies to feed its babies. Like morality in nature. Now, on the other hand, unlike humans, the hawk is not going to kill as many things as it can and destroy all its resources so that it can satisfy some kind of idealistic want, right? It's just living. Like that's what nature does. It chooses. When we say nature is choosing or best fit to survive, all we're saying is like, look, who survives and who doesn't under these conditions. All right, that was a lot of talk, but the bottom line is that that's called endosymbiosis. So remember that because that I've seen that on the OST, and it will probably be on your final. So the mitochondria is doing aerobic respiration, but in the cytoplasm, for sure, all anaerobic respiration is doing. It's not very efficient. So, it's a weird way of spelling aerobic. Aerobic, right? So anaerobic respiration is happening in the cytoplasm. Is that clear to everybody? So the part of respiration without oxygen is happening out here. And the first part of it is glycolysis. Glycolysis, the breaking down of sugar, is anaerobic. There's no oxygen being used here.
We saw that yesterday.
You did. You saw it in the video. You saw it in the packet. I talked about it in class. It's called glycolysis. And there's many steps, many enzymes, without which the steps wouldn't happen. There's a couple control points. The key here is that sugar is phosphorylate. The key here is that there's ATP put into, 2 ATPs go in to the reactions, and out of the reaction comes out, let me make this bigger because it's not. And out of the reaction comes 4 ATPs. And you have to invest in this 2 NAD pluses, NADs, and out comes 2 NADHs, carrying the electrons. FADH happens in the mitochondria, but that's good. I'm glad you remember it. So the 2 ATPs and 2 NADHs, you make here, so what's the net? What is the net gain? You put 2 in, it's called the investment.
And you get 2 out.
And you get 4 out, so what's the net? What's the net? Remember what is net?
6.
It's after you add and subtract everything. 2 is the net, yes. Because 2, you minus 2, because you lost 2 in the beginning, you invested 2. You gained 4, so you got 2 net. Very important business idea, net profit.
None.
By the way, you can make profit as a business person and be ethical. You can make profit and not hurt people. It is possible. But understand the system does not push you in that direction, right? So understand that. 2 NADHs. All right. So there is but the other thing that makes is this molecule called pyruvate. Yes, it's pyruvic acid. There's 2 of these are made. There's 3 carbons in pyruvate. 3 carbons. That's why there's 2, because in glycolysis, there's how many?
And 6, right?
There's 6 carbons in
So there's one glycol, it turns into
in a hexagon, right? It's a bad hexagon, but it looks more like a turtle. But anyways, so there's 6 carbons going in. There have to be 6 coming out to balance the equation. So each pyruvate has 3. That means there's 2 pyruvates, 2 ATPs, and 2 NADHs. The ATPs are used, but the NADHs are going where? Into the what's called the electron transport chain. Because NADHs are electron carriers.
Oh yeah.
The whole point here is to carry electrons to the electron transport chain that's in the membrane, the inner membrane of the mitochondria. This is known as the matrix. You know how you have the stroma in the chloroplast? We have the matrix in the mitochondria. But we still have a lot of energy in sugar, in pyruvate. In fact, most of the energy is right here. And these 2 ATPs are not enough to do all the things we need to do as human beings. So the pyruvate's going to come in, and I'm not going to go into the details. You saw the details yesterday and in the videos that I sent you. But pyruvate's going to come in, and the very first thing that's going to happen is 1 CO2 is going to come out for every 1 pyruvate, right? That carbon came out of the sugar. It started off as sugar, turned into pyruvate, and now it's carbon dioxide. Where's this gas going? It's going out of your lung. So that candy bar, that breakfast you just ate, if you go and burn it off, running around the building like maniacs, well, some of you run around all the time hiding things, but you run up and down stairs and such, you can burn all those calories. All that sugar you just ate, you just turned into carbon dioxide.
That's why you be breathing heavy.
That's one reason, but let's move on. So that's 1 carbon, but there's still 2 carbons left. Those extra 2 carbons go into something that has, get ready for this. I hate this fact. It goes into this thing that has 3 different names. It's insane.
Why 3?
I don't
Can
know.
you put it in one name on the final?
Please.
I don't know what name they're going to use on your tests that are not my final, but I'm going to use Krebs. I'll make sure I say Krebs. Krebs cycle, but it's also known as a citric acid cycle. It's like vitamin C, orange juice, yeah, vitamin C. Citric acid cycle, Krebs cycle, also known as a tricarboxylic acid cycle.
I can't spell that.
Tricarboxylic acid, TCA cycle. But we're going to stick with Krebs in this class. You just need to know that they name it 3 different things, and it's really annoying. So out of this comes 2 CO2s, again, for every 1 pyruvate. So now you got rid of all the carbon that you started with in pyruvate's gone in the form of carbon dioxide, all right, gas. It's gone out of the system. But what you're left with is more NADH, more, now you have this thing called FADH2, which is, all these are electron carriers. And the whole point is to go to the electron transport chain that we saw already, right? We already saw that in photosynthesis. So these electrons, the whole point of these is these are going to be going to the electron transport chain in the matrix. Some ATP was made as well in the Krebs cycle, but let's move on. So in this membrane is a bunch of different proteins in a row. I'm not going to go into all of them. It's not really, you don't need to know the names of all those, etc. What I am going to do, though, is let you know that we have these proteins. The very first protein is going to accept. It's going to take the electron, NADH, and it's going to make NAD plus a hydrogen ion. And what's going to do is as the electron goes in, I'm going to use a color here. As the electron goes in, it's going to take with it the hydrogen. Hydrogen is also going to, I'm sorry, I shouldn't draw it that way. The electron's going to be given here, right? It's going to come off of here and go here. And as it does so, hydrogen in blue, hydrogen ion, is going to go and follow it and get trapped on this side. So you're going from low hydrogen ions to high. So you need energy. Where are you getting that energy? The high energy electron. Where did we get that high energy electron? Sugar, right? From photosynthesis. We tore it apart from sugar. Where did photosynthesis get that energy for an electron? The sun. So the energy came from the sun. So very good. And so it gets transported down the hot potato chain, just like in photosynthesis. A bunch of different acceptors take it, and FADH2 comes in there and does it too. Other hydrogen ions go in, low to high. But then you get to the last. Now we need one last acceptor. What molecule is going to accept the electron at the end of the chain? Remember, these electrons have to keep moving. If you don't accept that last electron, if this last electron is not taken, this one can't be transferred, which means it all backs up. So what's the last electron acceptor in respiration? Respiration. Air, not air, oxygen gas. That's right. Oxygen gas is the last electron acceptor. It takes the oxygen, mixes with a couple hydrogens. Of course, there's two of these, right? Right? Two oxygens. So you need 4 hydrogens. I hope you're clear on that. So 2 oxygens. So you need 4 hydrogens. There's plenty of hydrogens out here. They're getting used up all the time. And so what you end up making is 2 waters, right? You do this twice. Obviously, you're going to do 2. Each one of these oxygen atom molecules is going to take 2 electrons, right? We're good with that idea? So it's going to do, so each one. So this is the only reason you breathe air. The only reason you need to breathe is for that oxygen to take that last electron. You've heard of cyanide? The poison? Yeah. It binds to this protein and it stops oxygen from getting taken, and it backs up and you die. So that's how cyanide kills you. It stops the electron from getting transferred to oxygen. By the way, you notice you're making water? Yeah. Where'd the electrons where'd photosynthesis get the electrons? Water. Yeah. The energy came from the sun. The electrons came from the water. Where did those electrons go? Water. To oxygen, then to water, right? So water, it starts with water, ends with water. 2 different processes. All right, so that's a summary of respiration and endosymbiosis. I hope that helped. And also we reviewed protein transport. Tomorrow we'll cover as much as we can, review that summary. Instead of making 4 ATPs, the mitochondria using oxygen can make 30 some odd ATPs here. How did it do that? It has all these ions, hydrogen ions on this side. It's very acidic. And it has in the membrane here an enzyme known as ATP synthase. And there's a number of them. This is just one example. There's a bunch of them in this space. It is a channel protein. It will allow ions through. It will allow ions through the membrane, but every time it allows an ion through, it makes a turn. It turns, and it takes ADP plus phosphate, and it makes ATP. A lot of it, as it gets the energy from these hydrogen ions going through from high to low. ATP synthase. An enzyme that synthesizes ATP. It goes through the membrane. This ADP and phosphate makes ATP. And where did it get the energy? From the battery. What battery? The battery it charged. How did it charge it? The electrons. Where did it get the electrons? NADH, FADH2. Where did they get the electrons? From pyruvate. Where did pyruvate get the electrons and the carbons? From glucose. From glycolysis. Okay? Does that make sense? Are we good? Because there's one other thing. What if there's no oxygen? What is the process? The process is called fermentation. Also, another form of anaerobic respiration that happens in the cytoplasm. There's glycolysis, glycolysis, and there's fermentation. If there's no oxygen, pyruvate goes into fermentation. And what you produce is CO2 and a little bit of ATP. And lactic acid in humans, and alcohol in other beasties. All right. That takes care of photosynthesis and respiration. Hopefully you review all this material and you're ready for your test. You have all the summaries and review materials you need.